So let's continue talking now about the modulation of cells features. So when nets are released in the body, they can interact with the extracellular matrix and also with the cells that are in the vicinity where they are released. So I'm not going to talk about the extracellular matrix, but there are some studies already showing that you have degradation of the extracellular matrix, mainly mediated by MMPs and also by elastase, that is some is here, that can digest some of the proteins from the extracellular matrix. And when they are releasing the extracellular medium, of course, they can trap microorganisms and kill some of them. But of course, there is a whole bunch of studies about the escape mechanism that different microorganisms evolved to escape net-mediated killing. But what I decided to talk is about the net effect in the neighborhood cells. So when nets interact with cells, and I'm speaking mainly of macrophage, they can signal in the cell, they can modulate the cytokine production by these cells and also the chemokine production. So the first study that we did in the lab, this was part of the Anderson PhD thesis that was finished in his postdoc at NIH. And we did this because it's very important for leishmaniase the differentiation of monocytes that are being attracted to the sunfly bite site in dendritic cells to present the parasite antigens and develop a protective immune response. So there is a very well-established protocol that you can differentiate monocytes, and I'm talking here about human cells. So you can differentiate monocytes that you purify from the blood into dendritic cells if you cultivate these monocytes with two cytokines, IL-4 and GM-CSF. So when you do this, monocytes are characterized by the expression of CD14 that is high in those cells, either in the percentage of cells and also in the MFI. And when they are cultivated with the IL-4 and GM-CSF, they decrease this CD14 and they upmodulate the CD1A. So this is what happened when you cultivate this in vitro with these two cytokines. If we add nets to these cultures, as you can see here, you have an increase in the expression of CD14. So you are not having this down modulation as you are expecting. So nets are reverting and also maintaining the CD14 expression in some cells. And this is really with a high MFI. And also they are not expressing so many, there are not so many cells expressing CD1A, and the MFI is really low. And here you can see the monocytes that are in culture, and when you add the nets, how they interact with these cells. So we are seeing here a net interacting with monocytes, and this net is labeled with antibody against elastase, and the cells are labeled with DNA showing the monocyte nuclei. Of course, this effect is mediated by net, but if you digest net, if you filter the net, or if you use the Leishmania supernatant, because these nets were stimulated with Leishmania, so we have the supernatant control from the parasite. You don't see the same effect. So what could be the nets could be doing in this culture? So Anderson realized that and demonstrate that actually nets are digesting the alpha chain of the IL-4 receptor. Actually, the elastase present in the nets are digesting the alpha chain of the IL-4 receptor, but it's not doing anything in the GM-CSF receptor. So if you measure the percentage of cells, the monocytes that are expressing this alpha chain of the IL-4 receptor, when they are incubated, when the cells are incubated with NET, you have a decrease in this expression, and this is reverted by using the elastase inhibitor or when you digest these NETs. Of course, we measure some cytokines, some other markers of the dendritic cells in macrophage, but it's important to show that the macrophages that are being differentiated in the presence of NETs, they are much more permissive to the parasites. If you infect those cells that are these monocytes that differentiate in the presence of IL-4, GM-CSF, and NETs, you have some cells that are much more permissive to the parasite replication than if you use digested NET or NET inhibit with the or using the elastase inhibitor together with NET, showing again that these effects mediated by neutrophil elastase. And here is just to show you that all the monocytes that are being differentiated in these conditions, the parasite replication, those cells, they all increase it. So what demonstrated that monocytes cultivated with IL-4 and GM-CSF, they differentiate in dendritic cells that express CD1A and produce TNF-alpha and IL-12, and all this is contributing to kill the parasite. Otherwise, if you add NETs to this culture, you decrease the IL-4 receptor, we increase some markers of M2 macrophage, we have more secretion of IL-10 and TGF-beta, and all this is the milieu that favors the parasite survival, parasite replication. So we are seeing that NET is modifying the differentiation of these cells. And this would happen also in the Leishmania infection site. Another study that was the Andres PhD and Barbara master. Barbara contributed with some results that are from her master dissertation, was the effect of NETs in the HIV infection in macrophages. So we did more or less the same thing. So we cultivate macrophages, human macrophages. They are differentiated in vitro, infected with HIV, and then we add NETs, as you can see here. The macrophages are stained with phalloidin in green, and the NETs are stained with anti-elastase, and you can see that they are interacting pretty well. And so if we treated those infected macrophages with infected with HIV just for 3 hours with these different concentrations of nets. Then we wash these cultures and we measure the HIV replication 12 or 14 days after this treatment. And even though we see a very important decrease in this virus replication. And this is nets stimulated by stimulated from healthy neutrophils, healthy donors. And here is nets stimulated by HIV neutrophils from HIV-infected patients. And we can see the same effect of this net in this setting, meaning that neutrophils from the patients, they produce nets, and those nets have the same effect as the nets from the healthy donor neutrophils. Also, they show in this work that the macrophages treated with this net, they produce hunches and MIP-1 beta, and these two chemokines, they are important to inhibit HIV-1 infection. Also, we look the presence of nets in the plasma from HIV-infected persons. And as you can see here, we have increased nets. And here we measure DNA associated with elastase. So we have an increase in this, the presence of this nets in the plasma of the HIV-infected person. So the other study that we did is normally people that work with net and they want to know exactly what is associated with this structure. They look proteins. So we decided to look if the nets could carry microRNA, and not just carry, but also transfer microRNA to other cells. So we took 6 different donors. We simulate these donors with 4 stimuli, I mean, PMA, leishmania, A25T, that is a transthyretin, is an amyloid protein. And we observe that there are some microRNA that are shared by all the nets stimulated by these different stimuli, some others that are being shared by 2 stimuli, some others that are just shared by the other 2. And so we identify different microRNAs in these associated with these nets. Oops. So we observed that there is this RNA, this microRNA, 142.3p, that is an important modulator of macrophage activity that was associated with nets that we are obtaining with this 4 stimuli. And we decided to study this microRNA in particular because it has already shown that it regulates phagocytosis. It's important in the differentiation of macrophage and dendritic cells and potentiates the innate immunity, and also interfere with antigen processing and presentation by macrophage and dendritic cells. So as you can see here, we measure by quantitative PCR this microRNA, and you see different levels of this microRNA in the nets. And this was the expression was correlated with the expression of PMA that is our control in these settings. And to show that this microRNA was really associated with NETs, we did this EMs. So neutrophils is stimulated with PMA, leishmania, transthyretin, and IL-8. And so you can see that you have DNA and you have the association of the microRNA 142.3p in all of them. And here is just the merge of these two staining, showing the colocalization of this microRNA and the NETs. But microRNAs could be associated with proteins or lipids carriers. So to see if they are associated with proteins and lipid carriers, we stained the NETs obtained by PMA in leishmania with Argonaut. That is a protein associated with microRNAs in general, and also one lipid, lipoprotein A1. And as you can see in those beautiful pictures, you see the NETs labeled in blue, Argonauta, that is in green, and elastase in red. In PMA, stimulated NETs, and also in leishmania, stimulated NETs. So you can see the green here. That's not so visible here. You just see some dots, but it's really labeled. And what is interesting, something that you need to explain later, the parasite is completely covered by Argonaut antibody, and of course elastase. And also we load the NETs with the apolipoprotein A1. So again, you have the DNA, the apolipoprotein in green, and the DNA histone, an antibody that recognizes histone, and you see the colocalization in PMA-induced NET, and also in leishmania-induced NETs. And you see the parasite is here, just to show that we really have a parasite. But in this case, it's not labeled as it is in Argonaut here. So you see the colocalization of the microRNA and some carriers that are not that are normally associated with these microRNAs. Well, we show that NETs are carrying microRNAs associated with protein and lipid carriers, but could these microRNAs carry the message to other cells? So we have this NETs, and you incubate these NETs with THP1, that is a macrophage lineage that we differentiate with PMA to macrophage. Normally, is the procedure that you do to have macrophage of this lineage. And then we incubate this macrophage with the NETs induced by PMA with during 2 hours. Then we wash and extracted the DNA from the this macrophage, extract the DNA down, and then we measure, we extract the microRNA from the cell, inside the cell. So showing that the macrophage interact with the NETs, as we already saw in other experiments, and this microRNA could be found inside the macrophage. But this was not enough to show that those nets carrying microRNAs are changing or doing something in the macrophage. So we measure two readouts. We measure PKC activity and TNF alpha production. So I'm just going to focus is different controls here, but when you incubate this macrophage with nets, like here, we have a decrease in the PKC alpha compared to control, not treat control. And then you have a scramble microRNA or inhibitor of microRNA that are exactly like the control. But if you put nets in those macrophages, they decrease the PKC alpha. And if you do net with the inhibitor of the microRNA, you revert this effect. And we have more or less the same in relation to TNF alpha. So we stimulate the cells with LPS, so you have some TNF being produced. If you incubate with just the microRNA inhibitor, it didn't change anything, but if you put nets, you increase the release of TNF. And if you put the net treated with this inhibitor of this microRNA, we increase much more the TNF alpha production. So showing that the microRNA is going inside the cells and is also changing some features that after this interaction, changing some characteristics of these cells upon interaction with this nets, and this characteristics are being inhibited when we put the microRNA inhibitor, specific microRNA inhibitor. But everything that I talked about nets until now, of course, it's a relatively new finding. Not the release of, well, the release of net when Ziklinski was here in the International Immunology meeting. We talked to him, and he was saying that a lot of people, mainly pathologists, they already saw the nets in different settings and always thought that this was an artifact. So he was the one that has the clever idea to find a function for this mechanism. But of course, there are a lot of controversy in the literature. So they did a meeting in 2017, if I'm not wrong, but this was published just in 2019, where they did a survey with all the, not all, but most of the participants, with different questions about what is known at that time about netosis. So based on this survey, they have classified the answers about different questions that they put in the consensus, these green ones, and the ones that doesn't have consensus at all. So it's very well established that we need to standardize net research. There is a role in immune defense and inflammation, because nets could be inflammatory, and of course they are playing some role in the defense, as we saw in some infections, for instance. Nets can aggregate, as we showed in the work by Periplaneta Americana, but you are going to see also nets aggregation tomorrow in the talk by Martin Herman. The morphology of net is also an agreement, a consensus, and the triggers of net formation. Also, there is not a lot of controversy around. The origin of net is something more questionable, because some people believe that it's net from the mitochondria, but many others. Actually, the Hans Simon is the one that claims that the nets from eosinophils and even some stimuli from neutrophils, they release nets from the mitochondria, but some others don't believe that. And the whole of peptidyl arginine deaminase 4. Ziklinski was one that's not, that doesn't think that PAD4 has a role in mitosis. The kind of buffers and conditions that were used for net purification is also a big discussion. The differentiation from other forms of cell death, as I show you, there are many cells in different conditions that can release DNA to the extracellular medium, but this doesn't not mean every time that this is a netosis. So because of that, they are asking and suggesting not to use the term netosis, but on the contrary, to use just net release or net extrusion, but not netosis, because netosis is not very well delimited what it is netosis, what is just some net release, some DNA in the form of a net that is being released. And it's more or less like the X that are being released by cell types. So there is a lot of controversy, because also there is a lot of cells that we know now that are releasing DNA, but not could not be completely characterized as a netosis. So there are some controversy in the field. And finally, the association of nets with disease. This could be a whole class, but I'm going to show just some few slides. So net release could have a pro-inflammatory effect or an anti-inflammatory effect. So when they have a pro-inflammatory effect, you depending on the amount of net release and how quickly and effectively he is cleared, it is a source of different antigens. So, for instance, in lupus, you have many problems with net release. So sometimes the nets are not cleared quickly by the DNase. Some patients have some defects in the DNase, so DNase is not working properly to digest the nets. And some others, they produce a lot of, well, all of them, they produce a lot of antibodies against DNA. And this is a hallmark of this disease. And sometimes these DNAs bind to the structure, and so they hidden the access of the DNA. So you have this big mess of nets, antibodies, and activation of complement and many other molecules that are involved in the inflammatory properties of nets. On the contrary, in some other settings, we have an anti-inflammatory effect of the net release. And this was done and observed, for instance, in cholesterol crystals, where you have nets that are aggregated in this around these crystals, or sometimes around pathogens. So you have aggregated nets, and these nets aggregated, they can confine the infection and inflammation, and also they can, it's already shown that they degrade some cytokines and chemokines, so decreasing the inflammatory effect of these molecules. So till today, we don't know if nets are good or bad. What we observe is that if you have an infection and you produce very few nets or any, you have, like in this case, an infection with, this is a person that was infected with Aspergillus nidulans, if I'm not wrong. So all these red spots are PET scan, where they show the dissemination of the infection here also. And then they did a gene therapy, where they transfect one of the components of NADPH to this patient. This was a chronic granulomatous disease patient. So they did the gene therapy, and so they restored the ability of neutrophils to produce nets. Of course, not nets, also there is host production. And so the infection was almost, in this time, almost, and the red dots are, the red arrows, sorry, they disappear. So curing the parasite. So there is a balance between nets. So few nets in some cases causing disease, because they are needed to contain an infection, for instance. But sometimes too much nets are also a problem, because then they start to have to affect tissue. They are a source of autoantigens, and they can cause autoimmune disease. So it's a balance that sometimes we have a lot, and then it's not so good. Sometimes you have a few nets, and then you have problems too. It's something that needs to be balanced depending on the disease or the infection or anything. So depending on the nets are being released, we can have organ injury in all over, in any place where they are being released. So we have already described nets participating in Alzheimer's disease, in stroke, in rheumatoid arthritis, in gout, glomerulonephritis, hemolytic uremic syndrome, ulcerative colitis, preeclampsia, as you already saw, some not exactly related nets, but they are involved too. Placenta and endothelial injury during pregnancy. In the heart, atherosclerosis. Diabetes is not exactly like this, but vasculopathy associated with diabetes. In the vessels, thrombosis is a big problem. Tomorrow we are going to see that gallstones in gallbladder, they are, NETs has a lot of role in this. We are going to see the nice paper published by Martin Herman group. In the liver also you have some disease, and lungs now is the big hit, because COVID-19 induced a lot of NETs in the lung, and this has a problem because it's toxic to the endothelial cell and also to some pulmonary cells. So you have a big inflammation, induced not only by the virus but also by the cytokine storm, because you have so much cytokines being produced, and some of those cytokines are NET inducers. So lung is a big problem, and before we knew that they are playing some role in cystic fibrosis, lung infection, acute lung injury, asthma, and chronic obstructive pulmonary disease. And in this disease, it's interesting because it's already been used. I spray with DNase that is helping to digest the material full of NET that are being produced in this disease. So here is a list that's far from being complete, but you have NETs involved in disease, in infectious disease and non-infectious disease, most of them in autoimmune disease. So experimental dysentery, fasciitis, mastitis, blood sepsis. Sepsis is a big problem with NETs because they are being produced inside the vessels, and so they are being adhered to the endothelial. So you are having some endothelial toxicity, and it's a big problem. Malaria, tuberculosis, leishmaniasis, that I'm going to show in a minute, fungi, aspergillosis, otitis by some bacteria, virus, Eimeria, that is a cousin of malaria, respiratory disease, lung inflammation, pneumonia, enterocyte damage, and as I told you, COVID-19 caused by SARS-CoV-2. That is the big hit that we are having in these times of pandemic. And non-infectious disease, spontaneous appendicitis, that is shown in the first paper by Ziklinski, Ziklinski group, that when they describe the NETs, preeclampsia, insemination, you have some NETs holding the horse spermatozoa, kidney in zebrafish, small vessel vasculitis, but you have also not only the small, but in the big vessels too, periodontitis, thrombosis, lupus, cystic fibrosis, transfusion, cerebrovascular inflammation, rheumatoid arthritis, atherosclerosis, psoriasis, and cancer. That is, you have a class by Robson. There's a lot of papers evaluating the role of NETs in cancer. This is a big issue too. And as I told you, we can see a lot of NETs in the leishmaniasis lesion. So here you can see the hematoma, that is the form of the parasite that's inside the vertebrate host. And those are NETs labeled in brown, stained with antibody anti-elastase. And as you can see, there are some patients that they produce huge amount of these structures in the lesion. Unfortunately, in this work, we could not correlate the amount of NET with some characteristics of the disease. We just found that if you have more mastigotes in the lesion, you have more NETs in the lesions too. And here we showed a NETs. One of these is labeled with anti-stone and the other with anti-elastase. And in green is the Leishmania parasite. The mastigote is in a nest of mastigotes in close contact with this structure in the lesions. And of course, there is a lot of groups that are studying therapeutic approach to target nets. And one that is already used is the DNA therapy that is being used in cystic fibrosis. Sometimes elastase combined with DNase to increase the solubilization of the sputum, the liquid material that is produced in this disease. But of course, there are some toll-like receptor inhibitors, calcineurin inhibitors, Hoss scavenger, because for all most of the inducers, they do with participation of Hoss, PAD inhibitors, and this other substance that target NET overproduction. So most of these are still in test, but DNA therapy is already being used, at least in cystic fibrosis. Of course, there are some problems that sometimes one of the problems with DNA digestion of NETs is that some microorganisms, they use these pieces of DNA to get the material for nutrition, or they have some enzymes to transform in adenosine that has some problems for, not for problems for the host, but not for the microorganisms. So you need to know exactly when you can use one or the others. And I'm finishing with this nice painter by Professor Julius Schaffstein that he did inspired by nets. So this is, in his conception, a neutrophil releasing nets in a tissue beautifully painted. So if you have any doubts, just put in the Google Classroom answer or send me an email. I guess you have my email already, but I can tell you tomorrow again. So I hope you enjoy, because I think that the mechanism is very interesting, and more than interesting is also beautiful, very nice. I like to see these webs. So see you then.